Hello, I'm Neha and I will be talking to you about our work on impact of multiple resource limitation of plants on herbivores. Before I begin, I want to point out that the slide numbers are at the bottom right in case you have specific questions while listening to the talk. We now know that plants are simultaneously limited by multiple resources such as water, nitrogen, and phosphorus. In its simplest form, multiple resource limitation or co-limitation of plants can be explained as an increase in biomass in response to addition of more than one resource. In this figure, plant biomass increases with addition of resource R1, R2, and a combination of R1 and R2, suggesting that the plant was co-limited by both R1 and R2 resources. Addition of resources not only increases plant biomass, but may also change plant tissue concentrations and ratios of resources. For example, addition of nitrogen has been shown to increase both plant nitrogen concentration and N to C ratios, which may have consequences for other trophic interactions. Our current understanding of resource, plant, and herbivore interactions suggests that increasing resources should lead to faster plant growth and higher plant biomass, resulting in higher herbivore abundance. However, this may not be true for all resources. The hypothesis, firstly, assumes that all resources function similarly. This is untrue because we know that resources such as light have a very different impact on plants than nitrogen or phosphorus. Secondly, it assumes that herbivores respond to the same resources as plants. This is also likely to be false as there are several examples of resources that limit plants, such as phosphorus in terrestrial systems, which is not known to directly limit herbivore growth. Alternatively, plants are not limited by sodium supply, whereas herbivores have been shown to feed preferentially on plants rich in sodium. Finally, it also assumes that herbivores mostly respond to plant quantity, but evidence suggests that herbivores can select diet rich in certain resources. Given these various possibilities of plant and herbivore responses to resources, we need a multifaceted approach to understand the impact of resources on herbivores. It is going to be crucial to identify potential links between resources and herbivores, especially with the projected variation in rainfall and other climate parameters which will affect water availability. Moreover, the continuous increase in anthropogenic inputs of nutrients over the past few decades is also likely to contribute to widespread changes in plant abundance and distribution globally, which may have downstream effects on other trophic interactions such as herbivory. Being able to predict the responses of herbivores to different types of resources may aid in identifying areas prone to habitat degradation or pest outbreaks. Therefore, we develop a theoretical model to understand the influence of multiple resource limitation of plants on herbivores. We will then confront the model predictions with some existing data. For the model, I will first describe the different categories of resources that we consider, followed by the basic structure of the model. I will then discuss a few potential scenarios of herbivore response to plant biomass and resources, and end with model predictions and supporting data. As I mentioned earlier, resources differ in their impact on plants and herbivores. Therefore, we decided to define three broad categories of resources which can describe a vast majority of resources. Since most life on Earth is carbon-based, we assume that one of the resources must be carbon or a surrogate resource for carbon assimilation, such as light or water availability. These surrogate resources, abbreviated as SRC, are taken up by plants and used during photosynthesis to fix carbon with some efficiency. The fixed carbon can then be used for metabolic function or for structural support. The second type of resource is a non-carbon resource that is limiting only to plants, such as nitrogen or iron in aquatic systems and phosphorus in terrestrial systems. It is abbreviated as OP for only plants. The third type of resource is one that is limiting to both plant and herbivore growth, therefore abbreviated as PAH. An example of this would be phosphorus in freshwater systems or nitrogen in terrestrial systems. Resources such as sodium that limit only herbivore growth can be discussed as a subcategory of the PAH resource as they behave mathematically similarly. Therefore, we develop a model with three limiting resources. The model assumes two consumer resource interactions, one between plants and resources and the other between herbivores and plants. The terms plants and herbivores are used broadly to define any autotroph and primary consumer respectively in the system, and therefore the model can be useful for both aquatic and terrestrial systems. The three resources, SRC, OP, and PAH, are supplied into the system at some supply rate. The surrogate resource to carbon may be light for aquatic systems and light or water for terrestrial systems. The other resources, such as nitrogen, phosphorus, or iron, can be assigned as potential OP or PAH resources depending on the system. Once in the system, plants take up resources at some rate depending on the plant biomass. Therefore, the rate of change of resources is defined as supply minus uptake by plants and mathematically described in this equation. Once in the plants, the resources are converted to compounds that can be used for other reactions. The internal concentration is shown by the lowercase r. Plant carbon content can either be allocated towards structural support or longevity, or for metabolic functions. An optimal allocation to metabolic function of carbon can be estimated by maximizing plant fitness measure. The labile metabolic compounds interact to result in cell growth and proliferation. The new growth can be described as a product of the different resources. This can be easily explained with an example from the terrestrial system, where carbon, phosphorus, and nitrogen are the three limiting resources. Phosphorus is usually present in the form of RNA and ATP, which aids in the production of nitrogen-rich enzymes during translation. These nitrogen-rich enzymes then act as catalysts to convert carbon sugars to other compounds required for growth and proliferation. If we treat the two chemical reactions as second-order reactions, we get the overall rate of the reaction to be proportional to the product of the concentrations of the different resources. These plants are then also consumed by herbivores. The per capita rate of change in plant biomass is the difference between growth and loss. The loss can be either due to herbivory or due to natural wear and tear. I would like to point out that in our model, any allocation to structural carbon also reduces natural plant mortality. Finally, herbivores that consume plants increase in biomass and eventually die at some rate dependent on predation and other natural causes. Therefore, the per capita rate of change in herbivore biomass is growth minus mortality. As you can see, I use a generic function HA to define herbivory. This can take several different forms based on our assumptions for herbivory. In its simplest form, we can assume that herbivores respond only to plant biomass. That is, any increase in plant biomass will result in a proportional increase in herbivore biomass. In addition to biomass, we can also consider a scenario where herbivores respond to tissue concentration of certain resources. For example, in terrestrial systems, some herbivores are thought to choose food based on nitrogen concentration of plants. Alternatively, herbivores may respond to plant biomass and a ratio of resources such as nitrogen to carbon or phosphorus to carbon. These responses may result in very different patterns of herbivore abundance. Therefore, we consider these different possibilities in our model to arrive at a variety of predictions. I will use a table to provide a brief overview of the variety of predictions. The table has herbivore response type along the rows and types of resources along the columns. As you can see, we found scenarios where herbivore abundance and consumption increase with increasing resources. But we also found scenarios where herbivores reduce in abundance in contradiction to our current understanding of resource, plant, and herbivore interactions. In addition to some of the more expected outcomes, our model also provides several novel predictions. Our model predicts apparent or indirect limitation of herbivores by resources that only limit plant growth, implying that any addition of these resources will increase herbivore abundance due to its impact on plant biomass. Consequently, OP resources can provide indirect subsidies for herbivore persistence. Herbivores need a minimum supply of limiting resources SRC and PAH for survival. For example, in terrestrial systems, this could mean the minimum thresholds of rainfall and nitrogen supply needed by an herbivore species. Our model predicts that increasing OP resource, such as phosphorus in terrestrial systems, lowers the requirement of other resources needed for herbivores to exist. This has important consequences for predicting changes in herbivore distribution in response to changing climate and resource gradients. Lastly, the other counterintuitive prediction from our model is the potential for herbivore abundance to decrease with increasing PAH resource, especially when herbivore growth responds to PAH concentration. This result occurs due to the fact that plant growth increases at a faster rate in response to PAH resource supply than the tissue concentration, eventually leading to a decline in plant growth due to density dependence. Slower plant growth then supports lower herbivore consumption, leading to a decrease in herbivore biomass or abundance. We confronted a few of these predictions with existing data to provide a proof of concept for our approach. We explored data available from other studies, from long-term research sites, and some unpublished data from our research group. The first prediction was on apparent limitation of herbivores. It suggests that herbivores should increase with resources that do not limit their growth. Our first example of this is from aquatic systems where primary productivity is limited by nitrogen and phosphorus. Daphnia are common zooplankton grazers in freshwater lakes that are strongly phosphorus limited but unlikely to be nitrogen limited. We analyzed a long-term data set on Daphnia abundance, dissolved nitrogen and phosphorus from northern temperate lakes LTER and found that Daphnia biomass was positively associated with dissolved nitrogen even after accounting for phosphorus availability in the system, as shown in this graph. The axes are both residuals, meaning that variance explained by P has already been accounted for. Our next example is from a nutrient addition experiment in the Serengeti grasslands. Nitrogen and phosphorus were added to fence plots in a factorial manner, and grasshopper abundance was estimated a year later. Even though grasshoppers are mostly nitrogen limited, we found that grasshopper abundance increased with phosphorus addition, suggesting an apparent limitation by phosphorus. Our other counterintuitive prediction was that herbivore abundance should decrease with resources limiting both plant and herbivore growth. We found some evidence for this in a long-term data set from Palmer NTER in the Antarctic. Krills are a group of marine grazers that are thought to be nitrogen limited based on their low N2P excretions. We found that krill abundance decreases with increasing nitrate and nitrite concentration as shown in this figure. The axes show only the residual after the effect of phosphorus and silica have been accounted for. We also found evidence for this in grasshoppers in terrestrial systems. In a multi-year study in the old fields around Cedar Creek, grasshopper abundance decreased with increasing soil nitrogen content. All these examples provide preliminary test of our model predictions. More rigorous studies from different systems are needed in the future to validate the model fully. In summary, we find that it is important to distinguish between the different types of resources while studying plant-herbivore interactions. Herbivore abundance can increase or decrease depending on the resource type and herbivore response. Herbivores can also show apparent limitation to resources that do not directly limit their growth. These findings have important consequences for herbivore exclosure experiments. The outcomes of these experiments may vary depending on the resource gradients and the type of herbivore response to resource concentrations and plant biomass. Additionally, we can also expand the model to include resources that only limit herbivores, such as sodium, and use it to understand the impact of these resources on plants under herbivory. In conclusion, our approach provides a general framework to understand plant-herbivore interactions by providing several alternate hypotheses that can be tested in both aquatic and terrestrial systems. These predictions, once tested, may be used for understanding herbivore response to global change. With that, I would like to thank my dissertation committee and lab members for their inputs in developing this project. I would also like to thank all scientists that have made their data freely available for the Palmer and Northern Temperate Lakes LTERs. Finally, I'm grateful to NSF for funding this research. The tiny URL link to the preprint of this work is tinyurl.com/mrl-herbivore. If you have any questions, please feel free to email me. I will be looking for postdoc positions soon, and I'm excited to work on a variety of topics uh, ranging from theoretical ecology, grassland ecology, and species interactions. I would love to hear about any postdoc opportunities. Thank you for listening.